Good afternoon. This is Dr. Dan Guerra from VerEvMed from the Pacific Northwest studios of our company. Let's get started with a new VerEvMed video lecture, shall we? All right. So today I'm going to talk about the immune response and some clinical pharmacotherapy, particularly immunotherapy, for specific cancers. In fact, it's a general discussion that can be applied because we're going to be talking about the level of T cell populations. So before we get started, I just want to tell you that I'm Dr. Dan Guerra, in case you haven't watched this before. That's me. That's my email address right there. There's our logo for our company. This is our official company website and email address. If you want to contact me directly, go to my email, or you can contact me through the company. Either way, you can talk about any of the videos I've made. And all those videos right now are on my Facebook and also my Twitter feed. I'm just showing you the Facebook page here. Go there. It's open to the public, and you'll be able to download any and all of these YouTube videos. And I think I've got over 30 of them by now. Surely I do. And so today, again, I'm going to talk about the immune response and how clinically at a level of immunotherapeutics, we are manipulating the immune response to enhance ablation of tumors in human cancers. So I'm calling this particular talk, Immunotherapy Needs a Boost from Treg Blockade. Now, for you to get what we're talking about, of course, I'm going to fill you in and a complete introduction of it. But just recall that T cells are one type of immune cells. They're one type of lymphocyte. You have B cells, also plasma cell, also known as plasma cells, when they switch to what kind of immunoglobulin they generate. And then you have T cells, okay, from the thymus. Now, T cells are really important in destroying tumors. They're really important in the acquired immune response. And you have two basic types of T cells, and then you have all kinds of subtypes. The two types are Tregs, or T regulatory, and what that means is they regulate what the other T cells do. And when I say regulate, I mean they tune things down. Because if you get too much immune response from T cells, you can go all the way from having no immune response and having an induction of tumorigenesis that can kill you in a very rapid amount of time, a very aggressive cancer, for example, with no T cell regulation or no T cell ablation of the tumor, all the way to having an autoimmune response where there's way too much T effector cell activity in populations in the bloodstream and in the tissues. So Tregs control that flow, okay? And they do it in multiple ways, but mostly by single transduction. All right, so that's the background you need for this, so let's get started. By the way, this is, of course, one of my favorite places in the world. This is outside of Moab, Utah, and I go there very often with my family. All right, so this is going to be a discussion of a paper I just plucked out of Science Signaling, again, one of my favorite journals. Came out in January, and so this is January still, 2018, so it only came out about, less than 3 weeks ago. Today's the 20th of January, 2018, if that matters. So let's get started. Tumors generate immunosuppression. They do it both locally and systemically. And they do it, of course, via biochemical signaling, like everything else in the cell is biochemical signaling. And that immunosuppression promotes T effector cell apoptosis, so it kills T effector cells. I just told you that those are necessary as anti-tumor agents from the acquired immune response. But at the same time, they activate the Treg cells. So they knock down the T effector cells, and at the same time, they induce T regulatory cells, which further causes a decrease in T effector cell activity. All right? So tumor infiltrating CD4 plus, FOXP3 plus, Tregs. Now those CD4, FOXP3, those are molecular signatures on the Tregs. When you see those molecular signatures, along with a lot of other ones, you're now defining a specific T cell population. When you have those two molecular signatures in their Tregs, those act as immunosuppressives in a mechanism that promotes basically tumor immune evasion. It's not invasion, it's evasion. Those same said Tregs corrupt the immunotherapeutic measures when treating aggressive human cancers. So at the same time that they can be ablating and diluting and inhibiting the natural immune response. When medicine goes in and tries to enhance the immune response, particularly enhance the T cell effector population, such as the cytotoxic T cell population, what happens is that the Tregs can block that. Okay, so that's a bad thing. So even when we try to do immunotherapy, these same said T regulatory cells can get in the way, and they can prevent the therapy. They can prevent the ablation of the cancer. So if you have an elevated ratio of Treg to TF, that's bad. It portends poor clinical outcome for patients suffering from any number of cancers, all the major ones: breast cancer, brain cancer, lung cancer, things like that, pancreatic cancer, liver cancer, okay, prostate cancer. So not a good thing at all. Very, very bad. Okay. So now one more thing you need to know before we really get into it. T regulatory activity is actually enhanced by a cytokine known as tumor necrosis factor alpha. So that normally you would think that cytokine is going to activate, because it says tumor necrosis factor, a subcellular population that's going to now cause anti-tumor activity. That is indeed what TNF alpha also does, but in Treg cells it does something it does something opposite, because it activates the Treg cells. You activate the Treg cells, you then block the T effector cells. You see. So that's a break on the system, and it's supposed to be there so you don't go to autoimmune. So let's take a look at this from a diagram. You got progenitor cells in the bone marrow, T cells, and you can go into these two major populations: naive T cells or Tregs. Tregs, when they bind to TNF, right, tumor necrosis factor, right, what happens is that they're going to block all these effector cell populations. Now, how do you get from naive T cells to Th1, Th2, or Th17, all really good, healthy effector cells, kill tumor, for example, and kill microorganisms as well? How you get there is by a subpopulation of cytokines that generate those subpopulations of T cells. So IL-12, IL-1, and then there's TGF-beta plus IL-8. Those are some of the cytokine signals that bind to the receptors on these naive T cells and then run them through further subpopulations. And all of these T effector cells have different nuanced effects on pathophysiological responses. Okay? And the important point here is that tumor necrosis factor receptor 2 binds to tumor necrosis factor alpha. It generates an activation of these regulatory cells and can block all those effector cells. That's all you need to know for right now. Okay, good. Now, this came from a Nature paper. This isn't directly from the paper we're talking about now, but the interaction of tumor necrosis factor with its receptor promotes a proliferative expansion, suppressive function, and phenotypical stability of Tregs. All three of those, not good when you're trying to get rid of a cancer. So take a look at this graph. This is the expansion of T regulatory cells on the y-axis, right? And this here is just showing, it's like a histogram, showing you whether or not you had TNF, a TNF agonist, or a TNF receptor antagonist. So you have TNF itself, that's the normal ligand for the receptor. You have a TNFR agonist. This is any number of compounds that are used to bind to that receptor, okay? And they can be antagonistic to TNF, for example, or they can, so that's antagonistic, or they can agonize it, which means they can activate it. So nothing here, none of these are added. Expansion of T regulatory cells is at this baseline. It's still above what's normal expansion, okay? Normal expansion. Now, when you add TNF, wow, look at the boost you get, right? From 20 to 40, right? So it's about a doubling of T regulatory cells. All you have to do is add TNF. Now, if you add an agonist, okay, a pharmaceutical agonist, you get the same effect. You get an increase in T regulatory cells. So that tumor necrosis factor receptor actually acts what? It acts more or less as a growth factor, right? It's expanding the Treg population. So it's acting as a growth factor, not just as pure activation. So that's an important issue here. Now watch this. You add an antagonist of the tumor necrosis factor receptor isoform 2 down here, and what do you get? You get back to being very low levels of expansion. So it looks like there's a cause-effect relationship. If you blockade that tumor necrosis factor receptor 2 with a good strong antagonist, you should be able to decrease the amount of Treg population expansion. So that's what it says here. Human TNFR2 plus reg cells increase in the peripheral blood of lung cancer patients and in the ascites of ovarian cancer patients. That's what we're looking at. TNFR2 is highly expressed in Treg cells. That's an important thing to remember, as compared to other CD4 T effector cells, for example, and to CD8. This is another subpopulation of T cells, which are very useful because these are the cytotoxic T lymphocytes, or the CTLs. Okay, so when you get TNF made, that's one thing, okay? It's going to induce a suite of events that are immune responsive. But when it binds to the receptor on the Treg cell, since there's a lot of that receptor on the Treg cell, you proliferate concomitantly more of the Treg cell over the T effector cells in both populations, CD4 plus CD8 plus. So once again, you got these Treg cells, you get naturally occurring. You get an expansion with an anti-CD3. Any of these ways you can get it, an expansion. Via interleukin-2 is another way. In fact, interleukin-2 binds to a specific receptor on the Treg cells called CD25. CD25 is basically a domain subunit for the interleukin-2 binding. Okay, so interleukin-2 binding. So what happens if you have a TNFR agonist? You get this enhanced proliferation of T cells, okay? And these are going to suppress all those effector cells. So these are all a nice lineage, very homogeneous population, and they're all going to do negative effects on the effector cells because they're going to blockade the T effector cells, which means they're going to inhibit the anti-tumor proliferation profile from the T effector cells. So you can also get if you add an antagonist, okay, if you add an antagonist, you're going to suppress this proliferation of T effector cell, of T regulatory cells, okay? If you don't have any further treatment, it goes back to sort of normal, right? And you have this heterogeneous Treg population, so you don't have this huge proliferation, right? So there is a temporal effect here, right? There is a half-life of how much of this T regulatory cell can be activated, even though it's expanded out by these various ligand bindings, such as IL-2 or rapamycin, okay? So that's an important thing to remember. Again, this comes from that Nature paper. All right, now, the amount of TNFR2, that's the receptor, present on the surface of Treg cells is associated with greater lymphatic invasion, a higher incidence of tumor metastasis, and a higher clinical stage and poorer response to treatment in patients with lung cancer and in acute myeloid leukemia. Okay, those are two general characterizations of what happens when you get TNFR activation via TNF or by a strong agonist on the Treg population. So TNFR is also found in several tumor cells. So that's the same thing. Includes colon cancer and Hodgkin's lymphoma. So it's all over the place. TNFR behaves as an immune checkpoint activator and oncoprotein. So obviously there are a lot of pharmaceuticals that are going to be targeting that TNFR2. That's the receptor, okay? So once again, just keep in mind what's going on here. So you've got any number of events that's going to cause an immature but still conditioned dendritic cell, okay? It's going to be receiving a series of signals, including some Treg control over it, and then any number of signaling molecules, TGF and all these interleukins, right? All these different molecules. There's PDL1, that's programmed death ligand 1, right? All of these cytokines, chemokines, etc., signaling molecules that are generated via this initial innate immune response are going to affect the Treg population, okay? Some of those Tregs are going to then inhibit T cell responses. They're going to suppress T cell migration to tissues and remodeling. They can also induce tolerogenic dendritic cells. So that's what you're seeing here. So it's like going back to the beginning. Tolerogenic means tolerating what? Tolerating the tumor invasion, right? Likewise, you're going to suppress the IgE and the IgG4 population. These are all going to be now, this isn't a tumor effect, right? This is going to be something like a bacterial infection, or maybe this is like some kind of immune response to a protozoan or to a fungus or something like that, right? So IgE responses, right? It's like parasitic infections and things like that. So, and you also get an inhibition from Treg cells to all these other really important innate immune cells, mast cells, basophils, and eosinophils. And of course, the eosinophils are going to be making an IgE. So all of this is bad news, right? But remember, it's not bad news to the whole organism, to the human body, because the Treg cells are putting the brake on the T effector cells. You got to do that, because if you don't do that, the T effector cells can overshoot their mark and they can induce an autoimmune response. They can kill too much tissue. And once that happens and it keeps going, then you might have a chronic autoimmune disease from chronic induction of inflammatory responses when you recruit the T effector cells. So that's why the T regulatory cells are very important. And you don't want to get rid of them, right? You just want to modulate them. That's a really important point too. Okay. So immunotherapy-induced tumor necrosis factor increases TNFR2 on Treg cells, which results in the expansion and activation, as we've been saying. By activating dendritic cells, the TLR9, that's the toll-like receptor 9, agonistic CpG oligodeoxynucleotide, or ODNs, okay, by activating DCs, the TLR9 agonistic CpG ODNs have the capacity to induce anti-tumor immune responses in mouse cells. So this is your normal case of events. Now, where do you get normally CpG oligodeoxynucleotides? Well, that's a nucleotide molecular marker. And as it turns out, certain toll receptors, now toll receptors are a big game changer when you're getting invaded by microorganisms, the toll-like receptors, but they're also activated by other kinds of stress events and pathological events, not just from bacteria, but also even from viruses and from protozoans and from parasites. But specific subclasses of these toll-like receptors are going to bind to these oligodeoxynucleotides. Now, where do you get this oligodeoxynucleotide? Well, you can get this from the partial breakdown of chromatin or from the genetic component of the microorganisms that are invading. A CpG island, okay, that's another whole story. A lot of CpGs are associated with epigenetic events, okay? So as it turns out, these CpG islands play multiple roles in facilitating signal transduction cascades that tune or turn down or up, okay? So I say tune, it can go up or down, right? Amplitude modulation, and also frequency modulation. You can shift from one class of subset of systems to another subset of systems. It can do that because these oligodeoxynucleotides are the product of expansion of that CpG island in promoter and enhancer regions of DNA. And when you expand that population, that expands the consensus network that allows you to methylate CpG islands, particularly on the cytosine residue. And when you do that, you can modulate via epigenetics by altering the expression of the genes that are being run by the promoter enhancer element. Okay, so I know it's a lot to say, but I just want you to keep in mind that the reason CpG ODNs work and why they're around as chemotherapeutics is because they're naturally occurring substances in the blood and in cells. So CpG ODNs promote the maturation and improve the function of professional antigen presenting cells like dendritic cells, while supporting the generation of antigen-specific B cells and CTLs. CTLs are cytotoxic T lymphocytes, right? Those are the T cell population you want, a subset of those T effector cells. Okay, good. So CpG ODNs are good. Now, intratumoral injection of CpG ODN also reduces the immunosuppressive activity of myeloid-derived suppressor cells. This is another good thing. So you have another subset of cell populations called myeloid-derived suppressor cells, MDSCs. These are different from Tregs, okay? These are progenitor cells, but they also are immunosuppressive, and you find them inside tumors, okay? They're kind of like stem cells in a way. So, but if you inject CpG ODN, you can reduce the immunosuppressive activity of those stem cells, which are like tumor stem cells in a way. Now, paradoxically, all this sounds good so far for the CpG ODNs. Yes, but paradoxically, the CpG ODN also induces Tregs, thus antagonizing the anti-tumor response because CpG ODN Yeah, guess what? It increases tumor necrosis factor, and therefore you know the routine now, you know the drill, right? The TNF is going to activate Treg cells, okay, because of the TNFR2, that receptor. So pharmacological blockade of that receptor could reinstate, you see, because the CpG ODN anti-tumor effect. Because the only thing that's making the CpG ODN not be a good player here is it's activating the tumor necrosis factor receptor 2 on Tregs. So if you can blockade just that, the ODN, the CpG ODN is going to be very functional good because it's going to promote the immune response. Okay, of course. So the science signaling paper we're looking at here thus examines the use of a TNFR blocking antibody. It's just called M861. I'm just giving you that so when you see it on the data, you'll remember. So that's a TNFR blocking antibody. Now, what's that going to do? Okay, if you block the TNFR, right, you're going to block the Treg activation proliferation. So that's a good player. So that's an immunotherapeutic, yes. Now, in combination with the CpG ODN pharmacotherapy, the paper is going to demonstrate that you can inhibit mouse CT26, this is a particular cell lineage, of colon tumor development. Plus the paper is going to examine the fate of mouse breast tumor when adding a blockade of CD25, which is another Treg activating receptor. I already told you what that was. It was the IL-2 subunit domain. Remember, IL-2 turns on Tregs, of course. All right, here is some really cool data. These are CD4 cells here, just total percent of proliferating cells, just looking at the whole total CD4 population. Again, you're looking here at the amount increasing on the y-axis. This is a histogram type of data set where you add interleukin 2, okay? You don't get much activity. You add TNF, you get and interleukin 2, so you're what? You're brush stroking both of those receptors on the Tregs. So something's happening to this total CD4 population. Boom, you're getting a massive increase in proliferation. But when you add the antibody against the TNF receptor, boom, you're almost back down to just the low level of stimulation from interleukin 2. Now check this out. This is the most important part of this. The percent of Tregs that are in that CD4 population is probably why you get this boost of proliferation. Again, IL-2, okay, there's some increase, right? There's TNF plus IL-2. The highest increase is here statistically significant against all other treatments. But when you add IL-2, TNF, which causes this boost of Tregs within the CD4 subset, it's a subset of the CD4 population, which is contributing to this first set of data. Yes. Look what happens. You blockade that TNF receptor, and you get back to being very low levels of expression. Okay, so that's what you want. So it looks like blockading, again, that tumor necrosis, just like we saw before, is a really good thing. Okay. Now, this is the percent of tumor necrosis factor receptor positive cells in the Treg. All right, this is telling us, is that protein there? What's the protein? The receptor protein, TNFR2. You just add interleukin 2, you got the baseline. You add TNF, look at that. You increase the amount statistically against all the treatments of the TNFR2, right? In association with IL-2, which is, again, brushing against that CD25 receptor, mediated activation of the Treg. But when you add, again, the monoclonal antibody against the TNFR2, wow, excellent stuff, bene, right? You get now back down of this decrease in TNFR receptor protein on the surface of the Treg cells. So obviously, not only is TNF activating, proliferating the Treg cells, it's also an autoinduction because it's increasing the amount of tumor necrosis factor receptors. You add more TNF, you put more receptor on the surface, okay? That's kind of how signal transduction cascades work. Now, after a while, an overabundance of TNF can actually cause endocytosis of that receptor. And that's what happens in things like drug addiction, okay? When there's such a strong activation of a receptor, like an opioid receptor, higher and higher doses of opiates, for example, eventually the receptor for those, like the mu receptor, is pulled back out of the membrane because of the high concentration of opiates. And that's why, this is a real aside, but this is just giving you a good, it's giving you an idea of what goes on with signal transduction, looking at ligand binding to receptors. That's why people who take opioid drugs need more and more of the drug for the same effect, and that's why they can also OD and die. Okay, because the less and less receptors, then they reach some threshold where there's just enough of the nasty drug, the opioid, that binds to the mu opioid receptor that binds and then kills, right? Destroys basically the breathing center for the brain. All right. Now, tumor size is decreased and survival is enhanced in mice treated with a combination of the CpG ODN and the tumor necrosis factor receptor 2 monoclonal antibody. Now look at here. PBS, that's just adding phosphate buffered saline. Tumor size after days after treatment, not a good thing. It gets bigger and bigger because PBS has no effect. When you look at either CpG plus, now this is IgG, okay, or you look at the M861 plus the ODN, which is not the CpG, but a control ODN. It's just oligodeoxynucleotides, but not the one that fires the TLR9 subspecies of the toll-like receptor, right? You get an intermediate amount of tumor size. It's better than just doing nothing, just adding phosphate buffered saline. But look what happens when you add the monoclonal antibody to the TNFR2, plus you add the authentic oligodeoxynucleotide, the CpG, which you know is going to fire that TLR9 and give you this intense response, right? Wow, excellent stuff. You get very low tumor size. And this is the same thing you see. These are the same treatments when you look at days after treatment of who's surviving, right? So who's surviving is out here. The mice that got the treatment of both the monoclonal antibody and the CpG ODN. See, they're surviving much later into. And these are days, but these are mice, right? So this would be like months or even years for a human. If you extrapolate, of course, you can't really just data, right? But showing you the survival is not so good with PBS for sure, right? They're dead at day 30. I mean, there's nobody surviving. You get a little bit of a positive effect when you're adding, either IgG or you're adding the monoclonal antibody to the TNFR2. But when you combine authentic TLR9 activating CpG ODN along with the monoclonal antibody that blocks the TNF receptor 2, this is where you get the highest amount of survival. All right. Now, breast cancer, same basic things happen here. Now, these things here, this TR75-54.7, that's just another monoclonal antibody to TNFR2. Don't worry about it. Just the name of it. It's a drug name, basically. This PC61 is a monoclonal antibody to CT25. Okay, remember that's the IL-2 receptor domain for CD25, that receptor protein. Same thing that happened before. Tumor size decreased when you block, when you block the TNFR and you block this other co-activating receptor. The more you blockade that whole system, the less tumor size you get. If PBS, you get no good effect. If you had just a blockade to the TNFR, you get some effect, but it's, arguably not very good. When you add just the blockade of the CD25, that gives you a little bit better, So same thing here with percent survival, just like we saw before. Most of the survival, now again, when you get way out at inoculation, these animals do die, That means that the effect does not last forever, Probably you need a new boost of this. So at any rate, what I'm showing you here is that when you blockade the TNFR and you block the CD25, you get more survival. And this is basically here. The median survival on days out after tumor inoculation, the best is when you blockade the tumor necrosis factor receptor 2 and the CD25, which is nothing more than another name for the interleukin-2 receptor domain. Right? That's the point. All right. So now let's do the dialectic. Right? We're going to put together this information. The dialectic, remember, just means that we're going to come up with a series of premises, and those premises then are going to arrive to a conclusion. So this isn't opinion. This is taking data and verifying it. So I call it a dialectic. I borrowed the term from Plato's philosophy, basically, way back where. All right, so immunotherapy that activates cytotoxic T lymphocytes, such as anti-PDL or PDLR. Now, those are topics of previous VerEvMed lectures that I provided, and they're all there on my Facebook page. So you can go back and look at those. But anti-PDL, that's program death ligand, or any antibody that's going to react with the PDLR receptor, what that does on T lymphocytes is it blocks a T lymphocyte from becoming apoptotic. So those are called checkpoint inhibitors, very common now in tumor ablation. So immunotherapy that activates the cytotoxic T lymphocytes, such as those kind of checkpoint inhibitors, may be and often are sufficient to target tumors and cause tumor ablation, anti-tumorigenic effect. Paradoxical activation of the Tregs, as we've been talking, along with TFs, such as the CTLs, can blunt the anti-tumor immunotherapy. Again, this is something we see, okay? It's out there in the evidence of the published literature. So, tumor necrosis factor receptor, interleukin-2, can activate Tregs. We also know that, even in the presence of an aggressive anti-tumor campaign. Okay, even in the presence, for example, of checkpoint inhibitors. So these would be like tumors that are becoming resistant to immunotherapy. And chemotherapy too, by the way, because I should mention that immunotherapy isn't yet considered the first line defense. Some rare instances there are, but normally you start off with chemotherapy, maybe even radiation, maybe even surgery. And then you go, when those tumors start to come back, or you worry about metastasis of some of the more aggressive tumors, then you might come back with the immunotherapy. So immunotherapy is not often used as first line, but usually in a sequence of events after chemotherapy, surgery, or radiation. But sometimes, I should argue, though, the literature is starting to show it as a primary. But at any rate, when you get these Tregs, you get this whole blunting of this whole response, and that's not good because the patient can die, right? The tumor progresses. So monoclonal antibodies to the TNFR and the CD25, which again is the interleukin-2 receptor domain, in combination with checkpoint inhibitors or chemotherapeutics or surgery or any number of combinations and sequential alterations of those drugs and therapies, and also like adding CpG ODN or even LPS to induce an innate immune response. This is normally done in subclinical, but probably would work in the clinical situation, although it's not done. Provide multifocal combination immunotherapy for otherwise resistant tumors. So this whole reason for this talk is to present to you this brand new science signaling paper, which is trying to hone in on how we can modulate Treg cells by blockading the tumor necrosis factor receptor, maybe the CD25 receptor, while still co-activating, maybe with immune checkpoint inhibitors or maybe just normal chemotherapy or normal anti-tumor proliferative drugs, then combinations so that you don't get that activation of the Tregs, so that you don't get the suppression of the T effector cells, such as cytotoxic T lymphocytes. So that's it. So entire paper, really excellent paper, by the way. I enjoyed reading it, like all science signaling papers. Very well done, very thorough. So I want to say goodbye now. That's me. That's my name is Daniel Guerra, and this is my email address. Please contact me for any and all criticisms or accolades about my videos. That's my Facebook page. Please go there and find all those videos. That's our company, VerEvMed. And there's our company's web address, www.verevmed. And there is our official VerEvMed email. All kinds of ways to contact me. All right. So let's end this presentation. And as I always say at the end of all of my videos, goodbye for now.